in the previous video we have understood about the cell cycle, and that too half. That means we have just understood about the interphase. Okay, so what are the different stages in interphase and all those things we have seen. So what we'll be seeing now is we will understand the M phase. What is M phase? What are the stages, sub-stages of M phase, or what are the different phases of M phase? And after that what we will do is overall we will understand the cell cycle. Okay, so now yesterday we have seen that if this is a cell cycle like this, okay, now this much time, that means if it is human cell, it will take 1 hour time for what? 1 hour for M phase, and the rest of the 23 hours for which phase? That is interphase. So this is at least clear in the previous video, interphase. And then also we have understood that again interphase is divided into 3 subphases like this: G1, S and G2. Also there is one more phase, okay, wherein the cells can enter one more phase called G0 phase or quiescent phase or quiescent stage, wherein cell is active, it is alive, it is metabolically also active. Okay, but what is not done there is division is not done. Okay, that we have also understood and we have done this much. Now what we will see is the M phase. Okay, what happens in the M phase? That is what we are going to see now. So let us consider this part now. Only we will see M phase. Now what is M phase? You know very well that M phase means what? If this is the cell cycle, here I will be just showing this. So this is M phase, I told you. Now within this M phase, what is going to happen? Cell is actually going to divide in this M phase. Yes or no? Cell is actually going to divide in this M phase. But M phase, to understand M phase again, what you need to understand is M phase is again divided into different phases. So what is that phase? So we will try to understand that. So this is M phase, and if I just show you this M phase like this, now this M phase is divided into 2 phases like this. Okay, this is the first phase, this is the second phase. What is the first phase now? The first phase here we call it as karyokinesis. We call it as karyokinesis, and the next phase is cytokinesis. Okay, now so what are the 2 phases here? These 2 are the subphases of what? These 2 are the subphases of M phase. M phase is mitosis phase. So mitosis phase again divided into 2 phases. The first one is karyokinesis phase and the second phase is cytokinesis phase. So what do you understand by the term karyokinesis and what do you understand by the term cytokinesis? And now what we are talking about, this you should not get confused. We are talking about mitosis phase. Mitosis phase. We are talking about mitosis phase. This is mitosis phase. Within this mitosis phase, we have 2 phases. One is karyokinesis and the other one is cytokinesis. First, note on this much. In detail, we need to understand that. So here, karyokinesis and cytokinesis. What is this karyokinesis and what is this cytokinesis? Now in cell cycle, here if you see, in interphase there was 2 or 3 phases. Now again, so we were talking about that. So already we know that it is G1, yes, G2 phases. And this 3 phases involves in interphase. So what happens here is cell is growing, DNA has replicated, again little bit of growth is done. Okay, ultimately all the proteins, all the enzymes, everything is ready for the division process. So now what will happen in M phase? Actually the cell should divide. This is what going to happen in the M phase. That is why we call it as mitosis phase or the actual division phase. So let us understand what exactly happens here. So here what happens is karyokinesis. What is karyokinesis? I will explain it with the help of a diagram. Now if you see, this is one cell. This is one cell like this. Okay, and then it has its own nucleus. Okay, and whatever the DNA doubling, everything has been already done. I'm just explaining you what is karyokinesis now. This is the cell, this is the nucleus. So first what happens when a cell needs to divide? First thing what happens is the nucleus will divide. So this nucleus, what you are able to see, this nucleus will divide first, like this. Like this. The nucleus will divide first. So the division of nucleus, why? Because this process is called karyokinesis. Why? Because nucleus is known as karyon. Karyon. What does the term karyon means? Karyon means what? Karyon means nucleus. So what is dividing now? Nucleus is dividing. First what will divide is nucleus. So because of this only, the first thing is karyokinesis. First thing what happens when the cell starts dividing, cell will not divide suddenly. First, the nucleus will divide. Once the nucleus division is done, it is not that easy. Nucleus division is not that easy. Nucleus is dividing means the chromosome present inside the nucleus is also dividing. All these things are happening. So, that will take place in different stages. So, karyokinesis is not a complete phase. In karyokinesis, we have different stages. We will come to that later. Understood what is karyokinesis? Karyokinesis means nucleus division. So here we will write that it is nucleus division. So what is karyokinesis? Karyokinesis means before the cell divides, first what will divide is the nucleus. So division of nucleus is called as karyokinesis. Then what will happen? Once the nucleus is divided, finally what you will be able to see is that now once the nucleus is divided, already the nucleus is divided into two parts. Now what will happen? The cytoplasm which is there here, the cytoplasm will start dividing like this. If it is plant cell, cell plate formation. If it is animal cell, furrow formation. Whatever it is. But what is happening here? Now actually the cell is dividing into two parts. So cytokinesis means what? So this part what we are showing, the cell is dividing into two. This is called cytokinesis. Now how do you remember the cytokinesis and karyokinesis? Karyon means nucleus. Cyton means what? Cell. So cell division, actual cell division, or you can also say that cytoplasm division. Here the actual cytoplasm is dividing now completely after these two processes. After this karyokinesis and cytokinesis, the cell is completely divided into two parts. Now you will be able to see this becomes one cell, okay, with the nucleus, and this becomes one cell with this nucleus, right? So this is one cell, this is second cell. So what happened now? This cell, after division, you will not find this cell, you will find only this one and this one. So this is what? This is mitosis. I mean the main stages of mitosis, wherein first what will happen? The first thing what happens is when the cell needs to divide, the nucleus will divide first. That is what we call it as karyokinesis, which happens in different stages. We will see that now only. And after that what happens is cytokinesis. The second process what happens is cytokinesis. So togetherly what it is? Togetherly this is mitosis phase. Is that clear? So in M phase, like how in interphase we have G1, S and G2, in M phase mainly we have karyokinesis and cytokinesis. Again karyokinesis is divided into different stages that I will be explaining now only, and we will see complete structure of cell cycle today. But just take down this much first. So I hope you understood this much. What is karyokinesis? What is cytokinesis? Okay, so we have completed M phase but not completely. So if it is done, we will see in detail now. In detail, M phase is divided into how many stages and all those things. Okay. Now, so as I already told you, within this, what? So M phase is divided into, first is karyokinesis and then cytokinesis. Now I told you that, let us write it here. This is M phase. What is M? M means mitotic phase. It is divided into two parts. One is karyokinesis and the other one is cytokinesis. And what is very hectic process here? Hectic process is karyokinesis. Why? Because the nucleus is dividing here. Nucleus division is happening in karyokinesis. And why this is hectic? Because nucleus के अंदर, nucleus वगैरह नहीं रहता है, chromosomes are there within the nucleus, and to divide that nucleus, first what we should do? We should condense that DNA which is present. So before the cell division, DNA is not in the form of chromosome. DNA is in the form of chromatin. Chromatin, that means thin fiber. Okay, just like threads. Later on, to divide this nucleus, first we should condense the chromatin to form chromosome. So much of work has to be done to complete this process, karyokinesis. So that is not an easy task. It is a difficult task. So there are again so many stages. So karyokinesis, if you see, we will write it first. Cytokinesis means cytoplasm. For your convenience, you can remember as cell or cytoplasm. Division. Clear? This much is clear. This is not a hectic process. This is very easy. Once the main thing is done, just dividing the cytoplasm doesn't take much efforts. But dividing the nucleus only takes more effort, and that also happens in different stages. Which are the stages? Just for today, we will just name it. Completely we will understand when I will be dealing with mitosis as a separate sense. So here, karyokinesis is again divided into different phases. See here. First is only four. So first one is called prophase. Just note it down for today. We are going to deal about this in detail. So the first stage is prophase. And then second one is metaphase. First one is prophase. Second one is metaphase. What about the third one? Second one is metaphase. Third one is anaphase. And the fourth stage is telophase. Is there any confusion for you people? I hope you people understood what I am talking about. See, however in detail we will do mitosis, but here to understand what are the different stages, because you should not get confused with this stages and the cytokinesis. Cytokinesis is not a stage here. Karyokinesis stages are prophase, metaphase, anaphase, telophase. 4 stages are there. Wherein within all the stages, all the arrangement is done in order to divide the nucleus. So once the telophase is done, the nucleus is completely divided. That means karyokinesis starts with prophase, karyokinesis ends with telophase. Once the telophase is done, then cytokinesis will begin, and that is not a big hectic thing. Easily cytokinesis, and that means cytoplasm will divide easily. So what you need to remember is, like interphase is divided into G1, S and G2 phase, M phase is divided into karyokinesis and cytokinesis. Within karyokinesis, again what you will do? First you need to understand prophase, metaphase, anaphase and telophase. All these things. Okay. So how will you draw the cycle now? If this is the first, you take down this much. Fine. Okay. So now let us see how to draw the cell cycle along with all the stages clearly. You have taken all these things? Shall I write this? Okay. Now complete the cell cycle we are going to draw, and we are going to understand how the cell cycle looks with all the stages without skipping any of the stages now. Okay, now it is easy for us to understand. Okay, let us draw a big one. Yes, this diagram is very much important. It's not a perfect circle. Anyways, center of the circle somewhere here. Not sure. Okay, fine. So, yeah. So first thing what we will do in this cycle, first thing what we need to do is, we need to first divide the two main phases. The two main phases are, first one is, actually the first one is interphase, and this is M phase. So where we will be writing it? Yeah. We will make one more circle to just understand the phase, okay? Like this. And then we will write, this is interphase. Okay, completely till here it is interphase. So within interphase, which are the stages? Within the interphase, which are the stages? There are three stages or three phases. Which are the phases again? Phases are G1, yes, G2. Okay, then, so this phase is what? This phase, what we will write here? This phase is M phase, no? That is mitosis phase. This is mitosis phase. So within mitosis, first what we will do? First we need to divide it into two. One is what? Cytokinesis. First is karyokinesis, and then, so this complete part is karyokinesis and this complete part is cytokinesis. So first we will write this cytokinesis here. Cytokinesis. And what about this? This completely it is karyokinesis, but we cannot write it here because within karyokinesis we have some other stages. So we will make one more line over here to just show that this complete part is karyokinesis, and I will be writing it here like this. Karyokinesis. This diagram is just to make you understand completely. If you see this diagram, you should not get confused with any of the stages. That is why. Now karyokinesis, within the karyokinesis, which are the stages? We will mark all the stages now. So the first stage, 4 are there, no? Like this. What is the first one? The first one is prophase. P-R-O-P-H-A-S-E. Prophase. Then comes metaphase. And then comes anaphase. And then comes telophase. Clear? So this is the cell cycle. This is the structure of cell cycle. The complete structure without any kind of doubts. Okay, anyways, one more doubt students will have, that where is this G0 phase? Somewhere in this G1 only you can show this G0. Okay. So suddenly as soon as the division takes place, it will not enter G0. No, some amount of growth will be happening, or even if it is not happening, once the division is completed, then only it is entering G0. So somewhere it will be somewhere in the G1 phase only. It is a side. That means G0 is not a phase within the cell cycle. It is separate. It is out of the cell cycle. It is not within the cell cycle. No need to worry about that. So what is the series here? Series is always remember like this. Always you remember like this. G1, yes. G2, and N. Just you remember this. However, let them give you whatever kind of confusion, even if they give you, they will just different ways they will give you. You need to identify that. So always remember with G1. G1, yes, G2, M. Clear? So this is the series how it moves actually. Okay. So I hope first you take down this one, zoom it, and you can. So if those people who are not understanding this one, once I'll just explain you. So this is cell cycle. First of all, this is the division from here till here. It is, sorry, from here till here. This complete set is what? This complete phase is interphase. Within interphase we have three phases again. This is G1, yes, and G2. From here till here, this phase completely. This is mitosis phase. M phase. So within M phase, the first phase here, from where? From this part till this part, this is what? Again, this is karyokinesis. And then just this part, little bit. This one is cytokinesis. And within karyokinesis, what you will mention? You will mention prophase, metaphase, anaphase, telophase. Clear? So this is the structure of cell cycle. Okay. Now there is one more important thing to understand in this concept. And that concept is about amount of DNA, and already we have done the basics, amount of DNA, about C and all those things. C, amount of DNA, content of DNA, which is denoted as C. We also have understood haploid, diploid cells. If you have suddenly jumped into this video and if you are not understanding anything, so all the links are also given in the description so that you can go directly, just you can click on that link and find out that videos, okay? But see here, what is important next to understand? This is the complete picture of cell cycle. We have completed the cell cycle here. We need to understand M phase in clearly. So mitosis, that is M phase is nothing but mitosis. So mitosis and meiosis will be doing separate one. This is all about cell cycle, okay? Now, there is one thing which actually students get confused about. That I'll be trying to explain. About content of DNA and all those things. Fine. Now if I write short form, I don't think so you people will have any problem. I'll rub this because it is too big. Okay. What I'll be doing is, okay, so see here, let us consider this is the cell cycle. Okay. Now I told you this is interphase. So within interphase we have what? We have G1 phase, yes phase, G2 phase, and this is what? This is M phase. Okay? Okay, fine. Now you know that G1, yes, and G2 are all interphase stages. Now what I'm going to explain you is that with the help of number of chromosomes, even if you consider human being or anything, we will see how it goes on in the cell cycle. Now let us consider this is one cell over here. This is one cell over here. And within the nucleus of this cell, this is a cell, within the nucleus of this cell, if we will consider it as human cell, you know that human cell, if it is undergoing mitosis, then this cell will become what? This cell is zygote, or any cell which has diploid number of chromosomes. Already I have explained haploid, diploid. So if you are not understanding, please watch that and come back. But again and again I cannot explain here. It will take much time. You know, diploid means two set of chromosomes are there. So human cell, diploid cell will have what? Will have how many chromosomes? It will be having 46 chromosomes. This is at least clear. Let us consider one of the body cell of our human body. It is undergoing cell cycle. wherein it is going to divide now. Okay. Now this is the number of chromosome. What is the number of chromosome here? 46 chromosome. Then what is the stage of this cell? Whether it is diploid cell or a haploid cell. This cell, if it is 46, it is having 23 pair. Means one set of 23 from sperm, one set of 23 chromosome from ovum or egg. So we will denote it as what? We will denote it as 2n. This is a diploid cell. Will you agree with me here? This is diploid cell. It is 2n. Now what is the content of DNA here? I am talking about chromosome as well. I am asking you the content of DNA. That also we have already studied. If it is haploid cell, the content is considered as C, 1C. If it is diploid, it becomes 2C. So what is the content of DNA here? So this is 2n cell, which is diploid cell, and the content of DNA is 2C. Here, where when it has just completed the division and the cell which is undergoing division, which is in the G1 stage. This is the condition of the cell. Now what exactly happens in the G1 phase? The cytoplasmic contents are increasing, or cellular organelles are increasing in number. Yes or no? All these things are happening in G1 phase. Now when this cell, all the organelles have increased and all the cytoplasmic content have increased, now the cell will move to which phase? S phase. Now, lot of confusion students have in this only. That is why I thought I should clear this. Now during this stage, the same cell, how many chromosome it had? Chromosome number is 46. Okay. Now what is going to happen in S phase? This also will show that already cytoplasmic content have been increased. So this is not, in S phase what are the two main functions? One thing is centriole will divide, and that is happening in the cytoplasm. Okay, that is happening and that is without any problem here. But the main function is what? DNA replication. DNA synthesis is happening here. So if it is 46 chromosomes here, still the cell is diploid, yes or no? The cell is what? The cell is diploid only, no? It is not haploid, it is diploid cell only. Now, during this S phase, what will happen? DNA replication will happen. Then do you think here 46 will become double and it becomes 92 chromosomes? Do you think that is going to happen here? No. From 3 videos also, this is part 3. The other 2 videos also I have told the same thing. Here what happens if it is a mitosis division, here 46 chromosome will not double. Chromosomes are not doubling here. What is happening? The content of DNA is increasing. Okay, as you know that DNA when it is, which is present in the nucleus, I have written 46. That doesn't mean it is having chromosomes here. Chromosomes are found only during this M phase. When it enters the M phase, there DNA will condense to form chromosome. Before that, here chromosome is found in the form of a thread. Now if you see this one, within this it will have just this type of DNA in the form of thread. But here also it is in the form of thread only. But why I have written this is to clear the confusion of students. So here also the cell is diploid. Here the DNA will replicate. That means still more convenience way to make you understand. Here 46 are like this, no? The 46 chromosomes what are present here, they are in this form. So what will happen in the S phase? Replication. Replication means what? Now this one will form its another half. When it was like this, what do we call it? This is also chromosome. After replication, what has happened here? Another copy of another chromatid has been developed. Now what do we call this? Do you think this is one chromosome? These are 2 chromosomes? No, we also call this as one chromosome only. So even this is one chromosome, even this is one chromosome. So what has happened is in S phase, this type of thing has happened. Not exactly like this, because chromosomes are found, condensation and all these things are done here. I am just trying to explain you, to convince you that what has happened, why chromosomes are not doubled here. instead of 46 during S phase, it will not become 92. Instead, what has happened is chromosomes which were like this has changed into this form. Changed in the form in the sense replication has happened. It has doubled the content of DNA. Then what will be the, here it was 2C. What will be the content of DNA? Is it the same? No. This was C. Sorry, this was 2C because this is from this cell. No, so this is 2C. Now it has become like this. Another extra copy is there. What it will become? So the content of DNA has increased to 4C. This is where children usually get confused with the concepts. They think that here in the S phase, chromosome number will double. Students think that here instead of 46 it will become 92, and then here while dividing it will give 46 and 46. No. Already I have explained you mitosis. What is going to happen in mitosis? Chromosome number will not double. Instead, centromere will divide. Yes or no? So centromere is dividing and forming chromosomes. That means here, now you saw that, okay, I'll just explain that also and make you clear here. So this what we had studied was mitosis, wherein we saw that like this. I am explaining with 2 chromosomes now. Mitosis, metaphase stage, chromosomes at the center. What will happen next? Next is the centromere of the chromosome will divide. What will happen again? Again I am explaining this. Make it clear. Centromere will divide. After centromere division, what will happen? One chromatid has moved towards one side, another chromatid towards another side. This chromatid towards this side, like this. All thing was within the nucleus. Now what happened? Cell will divide. Now what will happen? This will form one cell. This will form one cell with how many chromosomes? These type of chromosome which has lost its another sister chromatid. This will form one cell which is having again this type of chromosome with single chromatid. Yes or no? So these are the 2 new cells. Okay. Now these cells only, they are here. I mean the cells which are here will be having this type of single content of DNA. Okay. Since it is a diploid cell, content is 2C. Okay. Now in S phase what will happen? When this cell will enter G1 phase, cytoplasmic content will increase. When this cell will come to S phase, there what will happen actually? I mean before coming to this G1 phase, the chromatid will not be like this. Instead what will happen to this cell? Once the division is done, this cell means these 2 are in this stage now. Once they cross this, when they come to G1, when this is coming to G1 phase, what will happen to this cell is, first of all what happens is the chromosome will decondense and form this type of structure. Like this. Thread-like material. And during S phase what will happen? This content of DNA, totally it is 2C only, no, still. Now what will happen? This DNA, complete one, it will form its own replication and form another set. This also will form replication and form another set. Like this. During S phase, this is how the cell looks. Now, all the 46 chromosomes looks like this only here in the S phase. Now, this is S phase now. So, in the G1 phase, it was in the chromatid. In the S phase, it is looking like this now. Now what will happen later on? When it comes to M phase, that time it becomes like this here. So if I show you that with this cell only, the same cell, if this cell comes to M phase, during that time you will find that one part is like this and the new replicated I am showing with red. That doesn't mean this is another copy with a different thing. No, it is the same thing only like this. So I showed you with just 2 chromosomes like this, where the condensation will happen? This condensation of chromatin into chromosome is done here in this phase. That is, we are going to study in detail about mitosis. That time you will understand. What I am trying to explain with this thing is that to confirm you that when a cell undergoes mitosis, if it is a diploid cell, obviously diploid cell only will undergo mitosis in case of human beings. There are exceptions in other animals and in some insects, in some plants, there is exceptions, that also we'll see. But in this concept what I am trying to explain, once again I'll revise, just listen to me carefully. Whichever cell is undergoing mitosis, usually the cells are diploid, which will be having two set of chromosomes, so we call them diploid. If it is human being, diploid cell will have 46 chromosomes. When it enters when it is in the G1 phase, only the cytoplasmic content will increase, the cell organelles will increase. When this cell comes to S phase, what will happen? Chromosome number will be the same. But what happens is DNA has doubled. DNA has replicated and formed another copy. So there will be increase in the content of DNA from 2C to 4C. But if you consider it as chromosome, then there is no increase in chromosome. No, chromosomes have not increased. Even this is chromosome, even this is chromosome. So here actually if you see, this has converted into this form. That's it. So there is no increase in the number of chromosome. You will not consider it as doubled chromosome. No. This is chromosome, this is also chromosome. So what has happened? Chromosome number is same only. Even here in the S phase, chromosome number is 46. But what has happened? DNA has doubled. So DNA present in this chromatid and DNA amount present in this. Which is more? This only is more, no? So this is 2C. It will become 4C. 2C is the content of DNA in the diploid cell. 4C when it is in the replication phase. Later on what will happen? When it comes to this phase, G2 phase, all the proteins which are required for the division process are again produced in the G2 phase. After once everything is ready, then it will enter M phase. And M phase is nothing but mitosis phase. And that we are going to study in detail in the next video. So what I tried to explain here, I hope you people understood this. Okay, if any doubts, please let me know. If you have not understood, let me know about that. Okay, thank you.